Anyone have any questions for Mark? So we have on the schedule now just some sort of open time for discussion about anything and everything that you've heard today. So if people have thoughts and haven't had a chance to give a talk and identify their thoughts to the group, please feel free to do so now. And then also if anything during the day has shaken loose ideas for what might be community activities or resources that we should build in response to some of what you've heard, bearing in mind we've got a whole full day of great presentations to come tomorrow, please. And questions for Mark?
Okay. So I'm going to channel Judy Cho, who's remarked at one of our earlier meetings that we actually haven't seen a lot of new biomarkers in the last 35 years, since Judy started medical school, she says. And I think we have acute need for much better biomarkers, more kinds of biomarkers, biomarkers for more kinds of diseases. So I'd like to hear you now just talk for a little bit about where you think we could go most profitably in that area. I think it's an area of acute need.
Yeah, no. First, of course, is to recognize that there are multiple types of biomarkers, and the assays that you might want to have that might be suitable for use in a phase 2 intensive proof of concept trial where you may be more interested in establishing you've got target engagement may be different to those that you would want to be able to roll out in a phase 3 or in clinical practice and so on. So I think clearly there's a big opportunity around the protein QTL space, and I'm sure many people in this room have started to go fishing in those protein QTL databases with polygenic risk scores or individual variants of interest, and we certainly have, and have found some interesting things. We're still validating, and I agree, I don't think I've seen an awful lot published on those yet, but I think that's potentially a very powerful approach. But for other indications, we need biomarkers of toxicity, for example, and of course we have things like LFTs and so on, but we could probably do better in many of those areas. And so again, that's data that's often available in very large numbers, most notably from UK Biobank now, but from other cohorts. And so I think we can start to explore some of those data sets to see if we can identify other more subtle biomarkers of early toxicity that could be factored into drug development programs.
So I want to bring up to this discussion, sort of you showed this sort of landscape of different ways of dealing with the problem, and then progressively it becomes harder and harder and more targeted using animals and cells and targeted assays. So I wonder whether, so that's one way to do this, and that's very laborious and very expensive and takes a long time. But that essentially requires, it's kind of a proof. Every step you're looking for the proof rather than sort of a statistical association. An alternative could be, and of course we could build it at different levels, that at every level you use massive data, lots of reference data sets, you build correlations and associations.
Yeah.
It's not a proof, but there's enough accumulated evidence, and there's a probability that a fraction of those things will turn out to be true, yet you don't have any proof for any individual one.
Yeah.
Could that be an alternative you could do massively as opposed to targeted ones?
No, I agree. And I did frame that as simplistically linear because that was kind of the way I probably was thinking about it and how we were prosecuting our research. But some of you have seen me present that this much more as a cycle into which, as you say, you stuff multiple bits of large-scale data of various types until through some yet to be determined a mysterious process, a bell chimes and you say, yep, that one's ticked off and we can exit that. That's what I meant about metrics of success and validation. We need something in the field that gives us some sense of where we have sufficient confidence in a mechanism that we can tick it off. And of course, as we get more loci ticked off, that becomes a truth set against which we can drive innovation and the comparison method. So I think that's what you're saying. And
I'm not talking about also ticking off. I'm talking about a probability. So you cannot prove anything, but you just know that 30% of those will be true, and then the investment will be proportional to the probability of that being
Yeah.
No, you're a loud guy, but you take
Yeah.
the mic.
Okay. No, I was just saying that, just to repeat it, I was not saying that you accumulate enough evidence so that the evidence leads to a proof, so you can tick it off, but rather, if you know, out of these 1,000 things, you know, if 1,000 things satisfy a number of conditions, we know that 30% of those are, I don't know, you know, simplistically, if, you know, you have a co-localization between a GWAS and an EQTL, 30% of the times that's the effector gene. Now, that kind of condition where you don't have a proof for every individual association, but rather there's a probability associated with that, but it comes from massive data sets that do not require a curated experiment with an animal in that particular model, would that be an alternative to walk through the whole process until you get to the target?
Depends what you're after, right? Because if that 30% allows you to line up a pathway that is clearly overrepresented, that gets you additional information. So that 30% per locus could be translated to 90% chance of that pathway being involved. And that then, of course, but for me, going down the corridor to my therapeutic folks, area folks, and saying, 30% chance this is a real target, you know, do you want to take it on and think about doing a phase 2 trial? It's not going to go down particularly well, and I'll let you try and convince them of that. But so I think at some point you do need to turn it into a deal more certainty. And how much of that is generated through high throughput data and how much is hand-to-hand combat? I think we all agree we want to get as much of it as possible upstream, as I call it, in terms of large-scale data generation. So the amount of validation data that you have to generate through hand-to-hand combat is minimized and has a much greater chance of certainty.
Great. I mean, makes perfect sense, and I think that's one of the things we're talking about here is figuring out, are there general resources that can advance things down the line, and not simply expecting that there's a magical resource that lights up a place in the genome and says drug goes here. But Nadim, please.
Thanks, Mark. Of course, Nadim Sawar from ASI. I agree with everything you and Rob and Sally have synthesized. But earlier, Eric challenged us to be delusional, and I'm going to try and come up on that challenge. So could the ICDA inspire a whole new paradigm of therapeutic discovery that we're not thinking about currently? Because what you described is stuff we're thinking about currently, and there's two things that occur to me that we're not really doing at the moment. The first is trying to discover truly preventative therapeutics. So my focus is Alzheimer's disease. I'm not trying to develop a drug that will prevent someone from getting Alzheimer's who is truly asymptomatic. And through polygenetic risk scores and through other methods, there may be, and what Andrew just described, if you have a 66 million person clinical trial recall capability, could we envisage a time where the 50, 60, 70,000 person clinical trial required to demonstrate truly prophylactic efficacy could be envisaged? So that was one. You know, could the ICDA inspire or enable a future of prophylactic therapeutics? And the second, similarly, is at the moment, out with cancer, we still think about disease kind of in a site-by-site way. So I'm thinking about Alzheimer's or Parkinson's or FDD or DLB. But can we instead move to a paradigm where we think about cross-cutting mechanisms? So enhancing the ability of microglia to do this could be useful for all these patients. So I just like to encourage the ICDA to think about therapeutic paradigms that we're not currently thinking about, and kind of pan-mechanism and prophylactic therapeutics strikes me as two areas.
I mean, I'd certainly endorse that, and I highlighted one or two of them. We've heard some of them, but you know, we talked about at lunch about the importance of macrophage biology across multiple disease areas, including Alzheimer's and so on. I think that's probably a quite neglected area where there could be quite a convergence of interest across many groups, both in research and in pharma, to build foundational resources that would benefit a wide range of disease areas. So.
Great. Other thoughts, ideas? Please, go right ahead. They don't have to be questions per se, but this is just open discussion time.
So we know that most of variants are non-coding regulatory. So it's kind of easy to see how protein coding variants can be translationable from the pharma point of view. So what do you say we need to do to get those regulatory variants to the point of being interesting for pharma? How much evidence do you need for those? Because definitely you're not fighting the SNPs, you're fighting something they represent. So what do we know? We need to know pathway, a gene, or what is it?
The first step is to try and get confidence in the effector transcripts, right? Which, however much CQTL studies or high C or whatever you do, ultimately to me means some kind of perturbation study, ideally in humans. So, a huge amount of value in identifying causal coding variants in genes, knockout or not knockout, because not only does that help you establish confidence that you've got the right effector transcript, it also does one more really important thing, which tells you something about the whole body phenotype of that perturbation of that gene. Many of our GWAS signals are tissue specific, and they're not, those phenotypic spectrum of coding variants will not be absolutely the same. And to the extent that most of our drug therapies work across the body, although of course there are exceptions, we need to understand, before we get too excited about a GWAS effector transcript being a therapeutic target, to understand what the whole body phenotype of perturbation of that gene is. Ideally in humans, if not, we may have to go to other systems. So that's be my thoughts.
Thank you.
Judy, please, thoughts?
Can't hear you.
Just with these microphones.
Wonderful talk. So one of the most useful things mentioned a lot is pleiotropy, both expected and unexpected. In IBD, the joke is always we just think about using a drug that's been approved in psoriasis. And sometimes that works and sometimes it doesn't work. So how should ICDA think systematically about pleiotropy to completely square it off, to think about secondary uses and kind of from the pharma perspective, I mean, how do we plan for that?
I don't have a complete answer, but I mean, we heard a little bit of it from Ben Neal's talk today, and the ability to understand genetic correlations and use polygenic risk scores and so on to capture that. I mean, I do think we'll need to get down to the level of individual loci, because although polygenic risk scores are great, they are smoothing out a whole bunch of mechanisms at 100 loci or even 1,000 loci or more. And at any given locus where you might be using genetics as a way to key into a therapeutic target, there's no guarantee that that's going to behave like the rest of the crowd do. So sooner or later you have to get in and explore in a range of models what the consequences of perturbation are.
It's still not on.
Sorry. You're on now. You're on now.
Okay. Yeah, to follow up on what Judy was saying, actually one of the analysis that Haki Im's lab did with GCHEX data is looking at genetic variants that affect expression of multiple genes in cis. And we see that those kinds of variants, when co-localizing with GWAS, they basically have increased pleiotropy. And this applies both at the level of single tissues and also if you have basically genetic variants that affect gene expression in multiple tissues. So like some of the pleiotropy originates just like at the very low molecular level, instead of like that gene, like a single gene having sort of more diverse physiological consequences.
Molecular horizontal rather than vertical.
But I think these are great points. I mean, to me, I think one thing that we could do a lot better from the human genetics side is really cataloging the complete set of outcomes for a variant. And I think we could start by defining what does that actually look like, and then I think we can obviously start to fill it in more easily for coding variants, and these, of course, make sense to that they would have a broader set of impacts possibly. But as molecular studies begin to pin down individual non-coding variants, they could follow very much in step with an understanding both of their direct molecular consequence and then the broadest set of phenotypes across all biobanks and disease studies possible.
Okay.
So, this is Jonathan Pollack from NIDA. I think there's also an opportunity to use...
Speak up.
Sorry. I think there's an opportunity to use animal models to look at how, for genetic modifiers, particularly as you showed, Mark, in one of your studies, that PRS has an effect on the main effect, if I remember correctly in your talk.
Yeah.
So there
That
are
Mark,
like
that Mark.
Yeah, Mark. So there's the diversity outbred mice, and there's also other resources that are being created for rat, such as the rat diversity panel, hybrid diversity panel. Thanks.
Yeah, look, I agree. I mean, the field has gone much more human, and that's great because there's a lot of data. But I think we're now starting to understand exactly, in a better way, what the opportunities are to use animal models to recapitulate some of these phenotypes, and even to explore new ways of bringing data together. I mean, it's something we've been doing in the diabetes space of trying to take some of the affected genes that we think are operative in a human GWAS loci and do a reasonably scaled experiment in zebrafish and fly, and also obviously data from mouse as well, just to get a sense, because I don't think we have any sense of how often do those data all point in the same direction. And that's what I was really talking about in terms of metrics of success and validation. I think is a really important thing that ICDA can do is to gather data sets together from diverse approaches and run them up against each other, and ideally run them up against truth data sets where we can identify those to understand really what the performance of each of these is. Because clearly we can all do anecdotal science around, oh, this locus was solved with this method and this locus was solved with this method, but what we really need to understand is across the board, and also for each disease specifically, because it may not be a generic answer, which of the approaches that are going to get us where we want to get in terms of robust target identification with the most efficiency.
Hi,
Yes.
I'm Marilyn Miller. I'm the Program Director for Genetics of Alzheimer's Disease at the National Institute on Aging. So we have a large-scale sequencing project with 30,000 whole genomes in the pipeline, multi-ethnic, and about 10,000 are released for whole genomes and 20,000 are released for whole exomes. What we have now, we're at a place where leadership has asked us, so you've got these 55 pretty much okay places in the genome that are interesting. Can you please categorize these, tell us what's real, and tell us what function these real things have? And so we stood up a committee to start that process, and we realized we aren't even really sure for many of these things that have high value in GWAS, whether they are actually genes or if it's still a locus. Or is it, if it's between two genes, how do you pick which one it is? So this is a very difficult parsing process. It's extremely challenging, on top of which we have disparate functional data being piled on top of this. So we're looking very much to you to help us to get through this initial categorization process.
I think that's a lot of why we're all here, to try to figure out how can we, or what are the opportunities, the resources that are needed for this. And coming back to the last comment, I mean, there's obviously still a huge role for mice in biomedicine, but I think some of the opportunities that we heard from Ramnik, Aviv, the whole group earlier, would suggest to me anyway that some massively parallel screening in human cells might be the first line, and then moving on to animals after that. But that's just my view.
Yeah, but I endorse the question back there. I mean, that is one of the exciting things. I think we've all struggled with this variant to gene mapping, and that's fostered a lot of innovation around different approaches for solving that. And now we're starting to see the fruits of that coming through and the opportunities. And we within ICDA, I think we're perfectly poised to help to bring those strands together and understand what works, when it works, what the assumptions are, what the positive predictive value is, all of these standard parameters that we normally apply in other settings, but we simply haven't been able to codify or quantify until now. I think we do have an opportunity to do that.
Absolutely. I mean, I think this is the ultimate annotation of the genome that I'm sure many here have been envisioning for decades. And we need to look at all of these things as not individual experiments and projects, but as part of a whole that really does something transformative. Jesse.
Jesse Ignates from the Broad Institute. I just wanted to pick up on that thread of using CRISPR-based perturbations as a tool for learning about what human genetic variation might do. And we heard a little bit earlier from Jay and from Aviv about, and from Ramnik about examples of using CRISPR. But I think one general sense that I've had from the talks today is that there's a remarkable depth of expertise in this room about using human genetic variation as the perturbation. And there's a growing community that maybe is not as well represented in this room about using CRISPR-based tools and programmatically perturbing the genome as a way to learn about cellular biology. And I think those two communities are very complementary. Just to give one example, the ENCODE consortium has a new arm this year about using CRISPR-based tool to perturb regulatory elements in the genome. Jay presented on part of that data earlier today. And using, as part of the ENCODE consortium, we've been involved in analyzing that CRISPR-based data to learn rules of the regulatory genome that would allow you to then predict which enhancers regulate which genes, and therefore predict the effects of non-coding variants. And so I think the ENCODE consortium is winding down, but there's still this community of people who are applying these tools and expanding these tools, developing new techniques. I think it'd be really exciting to bring that group of people together with human geneticists to really capitalize this an approach of using genetic engineering to learn about human biology and developing predictive models that we can apply to human genetic data.
Okay, great. I agree.
Back? Great.
Can you hear me? Hello? Yeah, hi. So my name's Anshul Kundaje. I'm from Stanford. So I wanted to just put a question out there on computational models and hypotheses generated by these kinds of larger scale system level predictive models that will be coming out in the next few years, already are there. So there's been a lot of discussion about infrastructure for storing and sharing data. There doesn't seem to be a unified plan to have something similar for agglomerating hypotheses, millions of hypotheses generated by system level models, finding ways to reconcile these, prioritize them, discover new things from these hypotheses, feed them back to the experimental community to validate. And I don't know if it's too early to discuss these aspects. I personally don't think it's too early to discuss these things, but it might be useful to think about also that aspect of ICDA as an alliance, not just sort of a data-centric view, but also a hypothesis-centric view and a pathway from going to data to predictions to validation in a more like democratic way. Consortia, to some extent, are able to do that under single umbrellas, like GTEx has a really nice portal, and Code has as well. How do other communities that aren't part of these consortia take their predictions, models, and are able to easily integrate them into similar systems so that we have a unified resource of hypotheses?
I think it's definitely not too early. In fact, that's absolutely the kind of thing that's on my mind, is how do we tap into all of the expertise and all of the informative assays, experiments, and predictions that are going on to create a comprehensive annotation that everyone can use and refine over time. And I think this is it's very much in scope of what we should be discussing tomorrow in terms of creating resources for data that's already available, not simply focused on what other things we might do and how we share data that hasn't been created yet. So it's a great point, I think.
Yeah, I just wanted to put a call out for like the computational community to focus not just on hypothesis generation, but on reasoning engines around those, finding ways to prioritize and sort of reconcile millions of hypotheses. I think it's something that's underappreciated across the board.
Great. And let's have the last word from Rory before we move to pictures.
I was just wondering whether a premature belief that we know what's going on may actually be an obstacle to progress. And that, actually Francis Collins demonstrated that rather nicely in his talk in terms of what he would have predicted and how wrong he was in a number of areas. So, the idea that one should be looking at the genes, and actually when we do GWAS, we find that more than 90% of the associations are outside of the genes. If it hadn't been for the fact that you got all that data free, we might not have found that out. And a lot of the time when we, as reviewers, review grant applications, our job is to say why it won't work. And that may be an obstacle also to just creating the data and moving away from the kind of the old candidate gene or the hypothesis testing approach, assuming that we know what's going on and therefore we can go through this linear approach, and just actually move away from that and create a lot of data, as we did with GWAS, a lot of data of omic assays, a lot of data, not assuming that we'll be right in doing it, but there is a kind of the alternative assumption that goes on. Well, you have to be studying brain tissue in order to know what's going on in the brain. Well, maybe yes, but maybe no. And so I worry that in thinking about strategies of how to get from A to B, we think we know what we're doing. And it would be much better to assume we don't in our strategy.
Yeah, and on that note, I think we should.
Very good. Yes. Yes. You mentioned that we were. Yes.